Okay.
All right, let's get this night started. Hello, everybody. So, trust that everybody has one copy of the handout already. Anyone does not have one? Okay, awesome. Thank you. Are there any extras? Okay, come to the front, please. Thank you. Yeah. All right. So, welcome. So today we're just gonna go through a little bit of biological molecules. We get nutrients as well as enzymes. But before we dive into the content here, let's just get to know each other a little bit better. Okay, so this is me, right? So you can call me Teacher Jason, Mr. Pang, whichever you're comfortable with, right? But I have been teaching for about 10 years coming already. I hope I do not look like that, right? But previously I was with the Learning Lab. Have you heard of that? Right, so I was there for about 6 years, and now I'm with OVERMUGGED trying to reach out to more students like yourselves here. Outside of my teaching, right? I love being outdoors. So the background pictures here were taken in Australia, where I had the opportunity to climb and do a little bit of hiking. And in the middle fall, is something that I had the opportunity to combine my passions for climbing and teaching. So that was me teaching some climbing courses. And I also had the honor of meeting Jodie as well, right? I facilitated one of her climbing program at her school center too. Right, so that's just a little bit about myself, so that you are not just talking to a face, right? I am a person very much like you as well. So when we do get to start a class together, then I'll get a little bit better lah. Because today we are going to crunch through a very short session for the revision here. But moving on a little bit here, I decided to use the MRT map because the thing is a little bit more understandable for us than the actual map of Singapore. So we have a couple of places. We are over here. I see that Zheng is cheering for something. Where do you stay? Oh, okay, but they are in the works, not yet. Okay, right. So definitely you can follow the social media channels to understand more about our direction where we are opening more centers. Okay, but we are in Coburn 1 right now. So question I always like to share with any new faces for our classes over here is everywhere you go, no matter what kind of tuition center, even private tuition, what makes the lesson here with me different from other places? These are not O-level question, so just think generally first. What will make lessons here with me different from any other lessons you take anywhere else? Myrin, what do you think is different? Will be different here as compared to other places you learn with? Okay, well, thank you for saying that maybe there. Jada, what about you? What do you think will be different? So yeah, no difference What? But okay, thank you. Right. But yes, Marine is actually quite close really. I always like to share that no matter where you go, right, content will be the same because everything that you learn here will be catered towards what you need to know according to your school syllabus, which is from the ministry. Everyone does the same thing, right? Everybody can watch the same videos. YouTube is available for everybody. But the thing that differs, you will not get anywhere else. Is me lah. I will not be anywhere else. Very straightforward. So why am I bringing this to your attention? Is that as a student myself last time long ago, I found that connecting with the teacher is very important. If you do not connect, you cannot vibe and understand the teacher well, no matter how smart the teacher is, no matter how nice the place is, you will not learn properly. Right? So we have these events to open up to the public, to all of you, to give you the opportunity to see whether or not say you like me or don't like me, is whether or not you can vibe with my teaching style. Because as with all of you have different learning styles, every teacher has different teaching styles as well, right? So take this opportunity to see whether or not you can understand my language, how I bring the content to you, how I can show you that actually bio is not as difficult as you think it is, right? Okay, so what's part of my slides here? So also a perspective that I want to share with you here. As a student growing up, I never liked tuition, right? Because to me it was yet another thing to do on my already very busy schedule, and all of you are right now are even more packed than I was when I was a student here. So I want to offer you this perspective that learning with me here, if we do start a class together, is not something on top of your already busy schedule here. I am going to be able to help you to do whatever is necessary in the 2 hours I see every week here. And outside of it, you will do little to no additional work here. You will learn with me the best as you can. You will consolidate your understanding and cement your learning with me here. Outside of it, that's it really. Okay, so don't take this as an additional 2 hours that you need to spend learning, but as the 2 hours you need to spend learning. So it's just going to be school and tuition. That's it. Okay, right. So this is my perspective that I can offer you here. So for bio, it's very, very strongly about this first two. I want to share with you here. Okay, because unfortunately, for far too long, the word biology has been associated with this dreaded M word, memorizing. Thank you. Right. So it is my immense goal here. I want to move away bio and memorizing. I'm not saying that there's no memorizing involved. Definitely have. So allow me to share with you throughout this sneak peek today here how it can be easier to remember stuff than you think it is. Okay. Lesson forecasts especially important since we are Sec 3s over here because we have a longer runway towards your O levels next year. So lessons are always for you. Very straightforward. I can tell you right now, if you give me 2 hours to talk whatever I want, I confirm can talk one. I love the science. I will talk as much as I want to. But if it's not what you need for your exams, not what you need to learn, then there's no point in you coming to learn with me. Right? So when we do start a class together, I always tell my students here, tell me what you need, what topic you are doing in school right now. That's how we can calibrate and create a forecast that will help you learn best. Okay, so that's something I can share with you. So for the learning resources, okay, the first thing, very straightforward. I want to zoom into this one. Gabriel, are you happy to not have mere exams in school? Yes or no? You think it's a good or bad thing? Sorry? You choose one lah. No correct or wrong answer, just choose one. Good or bad? Good. Why good leh? Less stress, very good. Okay, so one reason here, don't be an exam, then less stress for me. Very good here. But we are missing one very critical thing that may us do give us the benefit of as well, knowing where you are in your learning journey towards your goals here. Okay, so here with OVERMUGGED, we also offer mock exams here. And if you look at the sentence over there, all the benefits, you get to know what topics challenge you the most. You get to know which concept in those topics you need to study more. But the grades that you get for those exams the school will know what. It doesn't affect your school grades. Okay, right. So that's something I can share with you. That is I think it's pretty exciting as well.
Yeah.
Okay.
Then last lesson, I thought there will still be more. There's three, but the first.
Okay, right. So just a little bit of thing here. Whenever we have big scale events, we will just bring food for y'all, right? So a happy tummy is a happy mind here, right? But I always say, don't join for this, join for the learning together. Okay. And yeah, this one is something I really like as well. So we are in the cold one, it's pretty new. This year then we start only. But over here, have a look over here. Maybe some of you are even doing it right now already, right? But maybe next time when you all start like really like chonging for your exams, you need a study space. At home, your dog, cat noisy, younger sibling noisy, your father mother ask you to clean the floor, clean the house. Right? This is where we can offer all our students are available, are welcome. Come to our classrooms as long as there are no classes or meetings happening there. Come in here to this beautifully done up classroom, very conducive for you to study. Okay, but that's a story for next time. Any general questions so far?
24/7 really big difference.
Text me first. Okay. All right. Talk about it next time. I can skip that. Okay. So take a minute to read this, please. You know who Mary Poppins is? Right. Okay. So just want to bring your attention to the sentence over there. So and everything that you do, whether you like it or not, there will be this small thing that you actually don't like about the thing. Right. So with regards to learning with me over here, hopefully I can help you find the fun in learning about bio here. And if you started off coming here with, I actually quite like bio already, allow me to help you love it even more. But if you come in here like, bio said, allow me to help you hate it less. All right. Okay. So with that, for our friends at home here, if you need to look for the soft copy, please scan the QR code there. For our friends in classroom, just give them maybe 10 seconds or so. You all can look through the paper first. Mingan, Jolene, Gabrielle, Janine, Mary, Sure. All right, let's go. So, biological molecules and nutrition in humans. So all the information you see in this set of notes are actually distilled from my full set of notes that will be available for to you once you sign up lah. So these are just very condensed versions. Okay. So this is where I will share with you my wonderful love for drawing as well. Okay, right. But you will find that I do a lot of annotations, so it'd be best if you can follow me along over there. I don't need you to do A-star level of drawing, because I also not A-star. Okay, so for our biological molecules, we're gonna start off with immediately we have our three basic biological molecules. Namely the first one being carbohydrates, thank you. What else? Fats and proteins, thank you. Okay. All right, so carbohydrates, fats, and proteins, I would like to highlight to you immediately. We have CHO, CHO, CHO. So all our biological molecules will have carbon, hydrogen, and oxygen. So this is a similarity for all of them lah. But similarities, too easy already. Let's look for some differences. I want to zoom into the first two, our carbohydrates, our proteins. Namely, the name carbohydrate. So I'm just going to use this word over here, carbohydrate. Okay, so if I zoom into this word carbohydrate over here, I can see that I have carbo over here. Then I have hydrate over here. So the carbo tells me, hey, what element is present? Okay, easy. This one's going to have carbon. Arista. When you see the word hydrate, what comes to mind? Water. Thank you, water. Right, and the formula of water. Arista, still you please. Formula of water is H2O. Thank you. And I want you to realize this is the first few things I can share with you, how to make things so much easier to understand. That those kind of eureka moments that Zheng just said is something I absolutely love here. Carbohydrates, as the name suggests, will always have carbon, hydrogen, oxygen. Hey, but fats also have carbon, hydrogen, oxygen. What sets them apart? The name carbohydrate. All carbohydrate molecules will always have H and O in the ratio of a water molecule. Then fats how? No ratio. That's it. That's it. Okay, so you can see over here, I have 12 hydrogens and 6 oxygens. The ratio appear as a you see that? Okay, so a very simple way to identify your carbohydrates based on the ratio of H is to O, based on the hydrate, the water. That's it. Okay? So therefore, this is what I was referring to. Hydrogen and oxygen, always 2:1. Okay, no matter what number. If let's say hydrogen, 46. Oxygen, 23. Oh, then that one is carbohydrate already. That's it. Okay? Alright. So then what happens if there are more than CH and O? This is where we have the CHO still comes with our basic 3 elements. Alright, now carbohydrate, fats, and proteins, I all have CHO. So that one doesn't give me new information. NPS. Nitrogen, sulfur, and phosphorus. Okay. So if the question states, oh, I got a biological molecule and got carbon, hydrogen, oxygen only. Oh, you know straight away, not protein already. But the moment they tell you got any of these 3 others, doesn't have to be all 3, If you got CHON protein, CHOS, CHOP, this kind, they got protein already. But usually N will be there also. Okay, so we will usually for proteins are CHON P or CHON S. Okay? So with that in mind, I've helped you identify very quickly one way to distinguish between our three biological molecules. Look for the NPS first. Have NPS, that's it. Your job is done. You found protein. Don't have NPS, then you look for the water. Have water, carbohydrate. No water, fats. Done. Okay? Can I? Okay. So now that we have learned how to distinguish them, let's look at how they are special. Okay, let's have a look at this word over here. Do we use this word in school? Recognizable, Okay, what does the mono refer to? What does the mono refer to? One. What's saccharide refer to? Sugar, very good, right? It is loosely translated from Latin as little sugar. So monosaccharide, I want to emphasize that is a K sound. Okay, it's not a CH sound. So a monosaccharide is one little sugar. Then I put two together then. Disaccharide. So this is my opportunity to take this little space over here at the top. Let us recap our monosaccharides. Megan, can you help me please? What are our three monosaccharides? Megan, what are our three monosaccharides? Any one of them? The simplest one. Learn maybe since primary school you exposed to it before already. Glucose, very good. Let's write that down. Glucose. Jody, can I have another one, please? Sorry? Okay. Lactose. Sorry, your name is? Si-en. Wait. Sucrose, I want to write here. Lactose, I want to write here. Cellulose, I want to write here. You give me three things, all not monosaccharide, Jaden. Thank you. Okay, if you're wondering why I write this kind of anyhow, there's a method to my madness one. So bear with me here. Okay, we come back to our monosaccharides, just ignore the other few words over there. Now, monosaccharides, they are the little sugars. If I tell you right now, all of them have 6 carbons, 12 oxygens, 12 hydrogens, and 6 oxygens. All of them have 6 carbons, 12 hydrogens, and 6 oxygens. Why are they different molecules then? Different bonding. No, still covalent bonds. Same covalent bonds. I think you're on the right track, but wrong word. How are they different molecules if all of them have exactly 6 carbon, 12 hydrogen, 6 oxygens each? That is what I'm looking for. Once more. What's your name again? Sorry. Michelle, what was the word you said? Arrangement. Alright now. Okay. So the idea is if I were to give all of you the same what's this? You know the Lego got like that one. If I give all of you 100 pieces of this Lego, will you all be able to build different shapes out for me? Yes, so that's the idea here. Okay, so the 3 monosaccharides that we learned all have 6 carbon, 12 hydrogen, 6 oxygens. Why are they different molecules? Arrangements are different. Thank you for that. Okay? Okay? What's the DS over here stand for? Disaccharide. Thank you. So they are made up of you have a question? Yeah. So disaccharides are always made up of 2 monosaccharides. Okay. We have 1 missing monosaccharide here. Disaccharide here. What is it? Try to remember names. Sorry. You are? Jadine, right? Jadine, what is the third monodisaccharide? Maltose, thank you. Okay, so why did I write it in this fashion? Okay, so all our disaccharides are made up of monosaccharides. Wow, then how? I need to remember so many things. What plus what give me this? What plus what give me this? What plus what give me this? Let me try right now. All of them. Okay, don't write first. You can just follow me along first. I'll give you time to write. All of them contain at least. Yes. All my disaccharides contain at least one glucose. So instead of remembering 1, 2, 3, 4, 5, 6 to give me this, I only need to remember 3 things. That's it. Okay, all my disaccharides contain, listen to this clue, at least one glucose. So the next thing I'm going to ask you is, hey, if maltose, I tell you, is the simplest disaccharide, what does it contain? That's it, 2 glucose molecules. Simple, right? So no fuss. Okay? Now, how do you remember glucose plus what gives me lactose? It's in the name. Look at that. So glucose plus what gives me lactose? Galactose. Oh. Galactose. You see that? Okay. 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 So glucose plus galactose gives me lactose. And therefore, we are down to our last one already. No other combination left. Okay, that's it, right? So you can either go down this way from maltose simplest, lactose point to galactose, then only left fructose or maybe like 3 years back, I suddenly got one girl, I was teaching this, then this girl shot suddenly, hey, sweet girlfriend. So, why you suddenly talk about my sweet girlfriend? Oh, sweet girlfriend. Okay, right? So if you follow my maltose, lactose, sucrose thing, straight away you remember glucose, galactose, and fructose already. Or if you want to remember your sweet girlfriend, also can. Okay. All right. So that's it. We have covered our 6 simple sugars already. Or simple carbohydrates. Jolene, yes, you're right. Monosaccharide is 1, disaccharide is 2. Okay, so I challenge you all. What is the molecular formula? It's the disaccharide.
So interesting.
That's true, right? So therefore, see?
Yes.
Say what you're thinking. It's not times 2, right? Let me see. It's not by math fail, Can see not? 6 times 2 is 12, yes. But the hydrogen oxygen, can you tell me what did I remove? What does that make? How many hydrogens did I remove? How many oxygens did I remove? What does that make? Water. Water. So this reaction, right? This reaction when I bring 2 monosaccharides together, this reaction is known as a condensation reaction. Because when I put two monosaccharides together, I have to remove one water molecule's worth to form the bond. This is how the reaction works, how to form the bond. It's like, for example, now you hold your black pen in your hand. Like if I want to shake my hand out, see, you must drop that black pen there. So when the bond forms, which you do not need to know right now, when the bond forms, the carbon atom must let go of its oxygen and hydrogen. Then you can form another person's hand. Okay, so simply this is how I can explain to you right now, but you don't need to go to that detail. Okay? Okay, this is the A-level thing. Okay, we're done with our simple sugars. Have I missed out anything? Very good. So let's go to our note that I am not referring to them as sugars anymore. Okay. What do we call them? I got monosaccharide, 1 little sugar. Disaccharide, 2 little sugars. Then how? What the others called what? Polysaccharide. Okay, but yes, right, the word poly means many sugars. Okay, so the first one was what Jody gave us the example of already. It was our, what else? Other examples? This one actually is the one your synthesis primary school one. Yes. Starch. Jadine. Absolutely, thank you. So this is also where I take the opportunity to tell you the polysaccharides are all made of glucose. They're all made of glucose. So Jody, how are they different molecules then? Absolutely, thank you. It's the same word there, just differently arranged. Okay, differently arranged. Right, so arrangement plays a very big part. They're all glucose molecules there. Will we be able to give a specific molecular formula for our polysaccharides? That one is try. Poly is many. It goes into thousands. So please don't go and multiply by 1,000. Okay, so you will not be atom, so don't need to worry. Okay, but the ratios still stand. Okay, all right, so with this, let's just do a quick recap on their functions. These two are similar to each other and different from this. How? What's that? Very good. Cellulose and starch are only found in plants. Whereas glycogen, wow, hey, don't shopping all silent your notifications, please. Okay, so this is one way which we can categorize them. Jolene, yes, you absolutely need to know all 9 carbohydrates are. And the reason why I didn't just blast it out and print it out for you is because if you will see 9 die, confirm, so much to remember. But if you track back to how I exposed you to them incrementally, not that difficult. Okay, not that difficult. All right, so there's another way I can differentiate my polysaccharides. This time is these two. No, wrong. These two. Storage. They're used for storage. Thank you. Michelle, can you help me? Then what is cellulose for? What is this? What am I doing to support? Oh, absolutely well done. Support. Where can I find cellulose? Of a plant cell wall. Why are plant cell walls so strong and rigid? Cellulose is the one that provides that rigidity in the cellulose cell wall. Okay? Jolene, yes, you only need to remember these 9 carbohydrates, okay? Don't worry. All right? Okay. At this point, just introducing to you the carbohydrates. Have there been things that you're learning for the first time today? Okay, that one fair. Condensation reaction, I would say, is the one that may be a bit more far away. What else? Jody, what about you? Have you learned? Are you learning something new here, or you all learned before already? Awesome, thank you. Okay, Ken. All right. So hopefully, what I have helped you see is to how to remember them more easily because it's more you link in the whole chain. All you need to do is remember the top part. The rest flows into your brain already. Okay. Awesome. Let's go. So, for carbohydrates, okay, the reason why I put it all in tables is because it's very easy to understand them and to see them immediately. So the function, the role in the organism is just there, very straightforward. Okay, if I were to ask you to look at this word over here, what organelle comes to mind? What organelle comes to mind? The powerhouse of the cell. Yes. Hey, always I must say this kind of thing, then you're, huh? Organelle. Okay, so you have the mitochondria. All right. And take note of the sentence that comes before this. All carbohydrates consumed are ultimately broken down into glucose. So no matter what form of carbohydrates you consume in the digestive system, you will always try to break it down into glucose first because that's the form the cell uses the most straightforwardness. Cannot break down.
Oh yeah.
We'll talk about that when we come to like nutrition in humans. Okay. All right. So let's see about carbohydrates. Go on to lipids now. Now, for your syllabus right now, it's interchangeable for you to use the word lipid or fats. Okay, both are the same for you right now, lipids and fats. All right. As I mentioned, no fixed ratio, and this is where I would like to channel my inner Michelangelo. Oh no, you're making me feel old.
Sorry. Only one showing face out now.
No, it's just an artist. Okay. When you see this drawing, you should be thinking of one glycerol backbone bound to 3 fatty acid tails. That is a fat molecule. Okay, the reason why I'm introducing this to you because if I were to share with you the name, this fat molecule is called a triglyceride. You don't need to know this, but why am I sharing this with you right now? So, if you do see this in your exam, it's not going to application already because I tell you a bit right now. Okay? What's the relevance? What if they tell you, oh, this other compound, it's called a diglyceride. Would it be easy for you to say that it is also a fat molecule? Right? Okay. So the thing I just did in my Sec 4 class just now as well, the content strictly, if you're going to tell me right now, hey, next time I don't think bio, I study this for what? Fair enough. The what cannot translate. I agree, can. If you don't take bio again, won't translate. This is the how. How do you think about this question? How do you find the answers in your head? That is what will come, that will follow you throughout life. So over here, if I tell you that triglycerides are fat molecules, then I tell you, oh, unknown compound X has a similar shape. Oh, then it's going to have similar functions, isn't it? Okay, think about that, All right, role in organisms, fairly straightforward, long-term energy storage, blah, 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 all these kind of things. One thing I want to engage you with is this point over here. Fats form the major component of cell membranes. So immediately, if you see this point, are fats important?
Yes.
If there are no fats, then you will not have cell membranes. No cell membrane, have cell or not? Then that's it, no life really. That's it, straight up, no life. Okay, so fats are important, but don't go and drink one bowl of oil. Okay, they are good and bad fats. Okay. Okay, last but not least, we have our proteins. The basic unit of a protein are the amino acids. Very straightforward. It's actually, I would say, the more complex one of the three amino acids because they have a huge variety of functions over here. If I were to help you understand the function of proteins compared to carbohydrates and lipids in just one word, proteins are the workers. They are the ones that do work. Look at the words I put under the rows here. They speed up reactions. They protect cells. They regulate processes. They move substances. Okay, so proteins are the most varied of the biological molecules, and simply because they need to do a huge variety of functions, they do a lot of things. Carbohydrates and lipids, I will classify them as resources. They are used, they are stored. Who use them? Proteins. Who store them? Proteins do. Okay? Okay? Questions? Before we flip the page. Awesome, let's go. So we have food tests. Okay, so in your syllabus, we have 4 food tests. Oh, just leave it here for our friends at home. So you are looking at the food test page there. In your syllabus, there are 4 food tests, one of which you have learned since P6.
Yes.
And here I would like for us to remember it in the full name, the what test for something. So it's going to be the iodine test for starch. Okay, so remember it in this thread here. So I did not put it down in your handout, so we're going to put it down together over here. The first test, so point it out with me here, is going to be called the iodine test for starch. Okay, now the steps are, like I said just now, I put in this table for you already. You all grow up already, you can read on your own, very straightforward. If I read off the table, no point. Let me help you understand this better. I'm going to zoom out here. I want us to tell me why the iodine test for starch is the odd one out. So let me give you a minute or so. Read through all 4 tables. Tell me how the iodine test for starch is the odd one out.
Because you only got one set.
No, it's not about the number of sets. It's not because it's the easiest also. No. Irene, do you have an answer? Expand on that, please. Yes. Yes. The iodine test is the only one, well done, Irene, they can be done on solid sample. Right? Well done, Irene. For all the other 3, it can only be done on an equal sample. Okay? All right, so let's go ahead and what's the name of test number 2? Benedict's test for reducing sugars. Okay, this is where once you're done writing whatever you're writing, let me your eyes on screen. I will head back to our carbohydrates. Okay, so Benedict's test for reducing sugars of our 9 carbohydrates. Which ones are reducing sugars? Ladies, I would love to hear your voices as well, please. Not saying I don't want to hear your voice, Jaden. Come, we are all learning together here, so feel free to lend me your voice in the classroom as well. Okay? Jody? Which else? Thank you for saying that. That was actually what I was hoping to hear. Okay, your reducing sugars are everything but sucrose. That's it. So instead of remembering 1, 2, 3, 4, 5, nothing wrong with that. I'm just offering you another perspective. Okay, well done, Jody. Right. So for your syllabus, once more, let me reiterate. You do not need to explain why, but you need to know that only 5 carbohydrates in your syllabus will return positive. Okay, the test is test for reducing what? Reducing sugar. So why do we ignore our polysaccharides? We don't even call them sugar at all. That's it. Yeah, you're right. Okay? Complex carbs. Okay? So from 9, immediately you cut away 3 already. So from the 6, only got 1 that is not. Well done, Jody. So once more, the phrase that is in your head should be all except sucrose. Done. Okay? Sorry? This is the part that too deep already. I need to draw this shape out for you. Okay? Okay? This is something for A's as well. Okay? Wonderful question. Thank you for asking that. Okay. All right. So, reducing sugars. Need to remember, reducing sugars is from the 6 minus sucrose, only left 5. Okay. Okay. So this is also a danger. You cannot say that it's a test for glucose because it's not. It can be any of the 5. Okay. Please keep that in mind. What else? Okay. Uppercase B because it's named after a person. Uppercase B named after a person. What is this reagent called? Okay. This is known as the. Let me find my highlighter. This is Buret's reagent or solution. So please do not confuse Benedict's and Buret's.
Or like that.
The reason why I put this over here is because there have been questions that do this. Okay, there have been questions to do this. Have you all done food tests yet, like the experiment? Have all? Do you all see the brilliant colors? What are the colors available for positive for Benedict? Brick red is the extreme. What are the intermediate colors? Green, I have green, yes. Orange, very good. Actually, these 3 colors only now, yeah. So from blue, nothing. You go to green, orange, brick red. What does this order tell me? Not the word level. Let's not use the word level. Intensity of color. Another word for amount of. Concentration. Yes, concentration. Okay, so in science we need to use the correct terms. Okay, concentration. All right. So again, we can also add this in over here from clear blue to brick red. What colors are we expecting? We are expecting to see green, then orange. Is it? Oh, yeah, orange.
So 3, 4 hours already. What's it called?
Yellowish orange. It's somewhere around there. Usually we can see orange. Okay, so until like that, okay, it tells me the concentration. Okay. And before I move away from this page here, I want to bring to your attention the word turns and remains. So when the question says, state your observation, predict the appearance, I want us to elevate our answer. So this is the time, exams is the time for you to flex. Show the marker that you know what you're saying. Oh, instead of saying solution is blue. What is it is blue? What are you telling me the meaning of the observation? Remains blue. This means it was and still is blue. So no color change. So therefore, what you're testing for, not there. If you say turns from blue to brick red, wow, you're telling the marker, I know the original color. I also know what is it right now. Okay, so I want to bring to your attention what remains and turns for all of our color changes there. Okay, so this applies to chem and physics as well. If you take physics then. Okay. Okay, for this one, I'm just going to zoom into the Benedict's one because this is where I share with you what Marin mentioned. We grind the solid sample with the mortar and pestle, then we add distilled water. Okay, So I want you to imagine now, I cry already, I have this paste, I add water. Do I test directly? What do I do to this mixture first? I mix it, then do I test immediately again? Take out But you're right, I need to filter. I filter the mixture. After I filter the mixture, do I test for the residue or the filtrate? Filtrate, Okay, how to remember? Residue remains on top. Filtrate flows through.
Oh, yeah.
So you test for the filtrate. Okay? So therefore over here, this is the so maybe you can sample it in over here. Add distilled water, then filter. Add distilled water, then filter. No, 是 sorry. 是 Okay, let it the words be enough to see. Okay, okay, good. Okay, excellent. Thank you for that. So therefore, if you are presented with a solid sample for these and these are do the same step. Okay. Okay. What else? Anything else? Okay, that's it. Any questions for carbohydrates, proteins, and lipids? Okay, thank you for asking that. Ethanol. Is it fit for human consumption? Ethanol is only one type of alcohol. In Sec 4 Chem, the last few topics you learn about organic chemistry, you learn about alcohols, yeah. Ethanol is the only alcohol fit for human consumption. So if it's the one found in your hand sanitizer, why cannot drink? Not about that. What? I heard something there. What is it? Was something mentioned? Michelle, you see something? No? Okay, it's not about volume. Wrong word. Huh? While that is true, there's a more pressing reason. First word that you said. Not quantity. Yes, it's just now the word there that you wrong one. Concentration. Thank you so much. Concentration. Concentration. Your hand sanitizer, what percentage? 70 over 90 leh. How do hand sanitizers work? They literally break apart cell membranes. You want to? Okay? That's how they protect your hand. What? It literally breaks apart the cell membrane. That's why don't go and like keep hand sanitizer everywhere you go. You will damage your cells. Yes. Breaks up the cell membrane. No cell membrane, no cell. If repeated exposure, yes, but like a bit only, never mind. Because why? After using hand sanitizer, feel cool, right? Why does it feel cool?
People the first day of the time.
No, the alcohol vaporizes very easily. When it vaporizes, hey, think back, P6, evaporative cooling. I'll come out of swimming pool, why so cold, right? Because the water on the skin gets heat from your body to evaporate. Your body lose heat, you feel cool. Why your hand feel cool when you have the alcohol in your hand? The alcohol gains heat from your body lose heat. So it will disappear very quickly. Don't go and like deep into. Okay? Can? So don't worry, just don't drink. You shouldn't be drinking anyway.
Don't look at me like
Okay, talk about alcohols next time. Okay. Okay, wonderful. Thank you for asking that. Let's move on to enzymes now. Enzymes is a very small yet big topic, I would say. Right? It's a content-wise very small, but application-wise plentiful. Okay, so first and foremost, I want to bring to your attention this word. Which one? Which one? I only got put like that one. Angeline, are you done? Right, for 3, 2, 1. Okay, let's go. Okay, complementary, because I do not want us to say similar. I do not want us to say identical. Right? The enzyme and the substrate are complementary with special emphasis that this letter, no, wrong. This letter is a letter E. If you spell it with a letter I, what happens? It becomes free, and enzymes are not free. Okay, please make sure you spell it correctly. Yeah, complementary. Okay, right. So we are also looking at the wonderful active site here. So just based on this, I'm sure you have heard of this, the log and key hypothesis, right? Okay. So if let me highlight this. Okay, so if you look at this idea, which one do you think is the log? Which one is the key? Is it the highlighter or the cat? Which one is which? Is that log? Is that key? Which one enters which? Oh, okay, sorry. Okay, everybody different. Okay, but this one is the one that goes in, okay, so the thing that goes into the other molecule, this is the substrate. The key enters the log. We see that the key enters the log. Thank you. So in this idea, sorry, let me again. So the key enters the log. Can this work? This talks about the, again, specificity. No, wrong, this one. Okay, about the specificity. It's not just touch lah, can already. No, when I ask you to shake someone's hand, you shake like that. Not shake like that, must be palm to palm. Okay, not just palm to palm. When I say shake the person's hand, okay, if I say okay, shake hands, you're not gonna shake the foot or something. Okay, so that talks about the specificity. Okay, right? So the key, which is the substrate, enters the complementary enzyme in its active site. Okay, so the most amazing thing about how enzymes work is to lower something that we call the activation energy. Can someone help me understand activation energy better? Who has done this in school already? What does activation energy refer to? Okay, close enough. Let's go. Let's write this down. Activation energy, sometimes referred to as in this symbol, uppercase E. Hey, the A got something different. Can you see? You know, penmanship. The A is a subscript. It's not here. Small a is here, in the middle. No, this is a bottom right.
What's the difference?
This one chemistry, you know? Hydrogen
Oh,
is that area. Okay, so if your chemistry you go and do like that, wrong idea. You see that? That especially for untidy handwriting or very small handwriting, they will fall into this trap. Make sure you all write it down properly. Okay? Okay, what is EA? Generally, we don't use the word number for quantifying energy. We just say amount lah. Okay. So just based on this sentence, do enzymes give their substrates more energy? The word that I need you to look at once more is lowest. Oh, what does it mean? Okay, so let me draw for you a hue. Point X, point Y. I need you to get from point X to point Y. Okay, but you need to climb this hill first. Because if I ignore the hue, then you just direct already what? So chemical reactions always have this hue. This hue represents the amount of energy required. Do you understand this graphic first? You want to climb a hill, from point X to point Y, you must unfortunately climb a hill first to get to point Y. So based on what we just highlighted about enzymes, what will enzymes do? They increase rate of reaction by doing what? That's what I'm looking for now. Lowering activation energy. That's the three words that we just highlighted over here. Lowering activation energy. So we actually do not. It's different from, erase this, then draw a low one. No, that's not what happens. The original remains. What do enzymes do? If this is a hill that you have to climb, right? The enzyme very smart. It creates another tunnel for you to climb. And this tunnel is lower. That's it. Are you confused? Okay, something I always say, confused, just say this is a place for us to be confused and then be unconfused. Which are doing okay? Yeah, don't know Masasa. So this is the new one. This is the EA with enzyme. That's it. So back to my question. Do enzymes give the substrates more energy? No, it just says that, hey, you know enough energy, you go there. You want to come here? Easier. Like. That's it. Okay? That's it. Okay? Okay, simply put, that's it. This graph is actually more properly drawn. Sec 4 Chem, you need to understand this graph in greater detail. For now, I just share with you the hue and energy. Okay? Righty, let's go. So, only when we form the ESC, only when the enzyme and the substrate hold hand together, then you can pass through this shorter. If you're on your own, you must go and try the higher one. That's the purpose of the enzyme. Okay? Hold hands can go better. All right, so answer me this. Why can the substrate enter the active site of the enzyme? They are complementary, Okay. Remember this? My next question is, why are the products forced out of the enzyme? Well done, Jen. Products are forced out of the enzyme because the products are no longer complementary. You can enter because you're complementary. You stay there because you're complementary. The moment you change shape, not complementary anymore. Forced out of the enzyme.
Why?
Huh?
Why?
Okay, why? If this thing, what's the name of the enzyme that breaks down maltose? Maltase. Do you know this? Not something new, you all recognize, Okay, so if maltose enters a maltase enzyme, what will I have now? I will now have two glucose molecules, which has additional what? What do we take out for this reaction to occur? Water. Now they got extra water. They take up more space. Cannot fit really. Not complementary.
Oh,
Okay?
yeah.
Okay. So therefore, you will not need to go into that amount of detail. But since you asked me, I try and help you understand. All right. So simply put, what I need you to remember is, why will the products be forced out of the active site of the enzyme? The products are no longer complementary. Okay. Okay. Now I want you to look at the enzyme. Compare before, ignore the during, and after. What happened to the enzyme? Okay, this again points to your definition of the enzyme, right? Remains unchanged, chemically unchanged at the end of the reaction. Okay? So enzyme action, that's it really. Okay, it binds to the substrate, provides an alternate pathway with lower EA, thereby increasing rate of reaction. That's all. Enzymes do not make the molecules move faster. They do not give the molecules more energy. How can we give the molecules more energy? Temperature. Alright, by now all of us should know, or today I will tell you, temperature is the measure of the amount of energy. The higher the temperature, the more energy you have. So if you look at point number 1, as I increase temperature, activity will increase. This is a very unique characteristic that I need you to remember. Every 10 degrees Celsius increase, rate of reaction doubles. But only to a certain point. What is that point called? You have the optimum temperature over here. The optimum temperature is where we have our maximum rate of reaction, maximum enzyme activity. So if at this point at 40 degrees Celsius, wow, enzyme activity is maximum. The enzymes are moving at their fastest. They are going around forming complexes, providing the lower activation energy pathway. What happens when I cool the reaction down? At this point, what word do we use to describe the enzyme? Using one word, we can use they are inactive already. Okay, they become less and less until they are inactive. That's what we're looking at. Okay, so below optimum temperature, the word I want you to remember is that enzymes now become inactive. I would also like for you to write down, very importantly, in open and close inverted commas, and never write it down in your answers. Never say that enzymes are asleep. But why am I writing this down for you here? Because when you're asleep, can I wake you back up? Can I? So therefore, if an enzyme has been cooled to inactivity, I can reactivate it simply by warming it back up. Very important point, because this is direct contrast above optimal. Open and closed inverter is dead. Proper word is denature. Can I bring a dead person back alive? That's it. This speaks of the irreversible nature of denaturation. When an enzyme denatures, that's it. I have lost the enzyme forever. It's as though I have chopped off your hand. You can never shake another person's hand. Okay? Okay? So this, please write down for me. This is denaturation. It's irreversible. Now we could bring in KPT, kinetic particle theory, to explain why the enzyme, kinetic particle theory in chemistry. Why would the enzyme denature? What is denaturation first? Yeah, so denaturation can be described as the irreversible change in the shape of which part of the enzyme? Always zoom into the active site there. Okay, so denaturation is the irreversible change or the loss in the shape of the active site of the enzyme. Okay, active site of the enzyme.
Right. If it's irreversible change, then wouldn't that mean you can do for another one, another substrate is complementary?
I drop off Henry. Can you shake another person's foot?
So it's not bad lah. But then how to
It's destroyed.
Oh, okay.
Okay, it's destroyed. Good question. Yeah, question. Okay, so denaturation in a way is destroy, but don't write destroy, denature. Okay. Okay, Ken, any questions on temperature? So our temperature right now very straightforward. Amount of energy. What is this? You may not have learned it yet in chemistry. But it's how what or what something is. Okay, let's go. pH refers to how acidic or alkaline. Okay. For pH, the same for both sides. So for temperature different, as you lower temperature below optimum, the enzyme has less energy, works more slowly, becomes inactive. But when you go above, wow, smashed, you lose the shape. But for pH, the moment you move away from the optimum, that's it, denature already. Because temperature is about energy. I give you more, you work better. Give you too much, you shatter apart. Okay? But what is pH? How acidic or how alkaline? I want you to think of it as the environment. The moment you move away from the optimal pH, enzyme say, you know what? I'm not going to do it anymore. That's it. A bit higher only, go on already. A bit lower only, go on already. Okay? So until you get to a better. Okay. Have you done acids and alkalis in chem yet?
What's that?
Lower sec. Okay. What ion is it that makes something acidic? H plus, thank you. Is a cation? Yes, fair enough. Okay. So therefore, it's about how much H pluses there are in the solution. Different enzymes like different concentrations of H plus. So let's say enzyme A can only function when there is 1,000 H plus ions. The moment you go to 1,010, oh, that's it. I'm not going to work anymore. Denature. Okay? For now, that's all you need to understand for this. You will not be asked to explain the exact chemical specifics. Okay? So you can see, temperature is a bit more forgiving, whereas pH is very, very restrictive, very narrow working range. That's why I mean that. Okay? Okay, questions on enzymes? Alright, so the third topic that we're going to look at today, why I clump all these three topics together is because it's additive. It adds on to the next. From biological molecules, the enzymes that break down those biological molecules to a system where it's breaking down first. Nutrition in humans. Okay, as always, right? Reading, you all are great at reading. So let me bring out some important points for you. Ingestion. Alright, so ingestion is where food enters the body, and we reserve this word for organisms with a mouth. Okay? Are you thinking of organisms without mouth? What organisms don't have mouth? Huh? That's why plant don't eat lah. They make their own food.
Oh,
What else? Organisms without mouths. Are they organisms? Isa? Did I say that right? Isa? Aisa. A minute. Is that correct? No, how do I pronounce the name? Sorry. Oh my goodness, I've got the R. Sorry, Arisa. Give me an example of an organism without a mouth. Yes. You say cell so close, bacterial cell lah. This cell can be considered organism, what? Cells that are part of a multicellular organism, not organisms themselves. Thank you, bacterial cell. Okay, yes, Sam, you're right as well, bacteria. So for here, ingestion is for must have mouth one. Okay. All right. Digestion. You all learn since primary school, very straightforward. When I taught this to my primary school classes last time, I always watch the magic school bus with them. There's no time to watch. Today to go through so many things. Okay. So the story for next time. For absorption, we learned that in the small and the large intestine already. Something new will be assimilation. Alright, please pronounce it correctly. Assimilation. Now, what is assimilation? Let me share with you two words. Generally speaking, assimilation describes how the body uses or stores a particular nutrient. Okay? So as I was talking about ingestion just now, ingestion enters via the mouth. Egestion exits via the anus. Okay? Very importantly, this is where we depart from what you all have learned from primary school. We do not call egested material waste materials anymore. Why? Urine contains waste material. Right? So now that we are coming to our side here, we are using more specific definitions to separate and distinguish materials here. Because is undigested food devoid of nutrients? Don't have. Undigested food, is it no nutrients anymore? So have. Then why we don't take? No, not enough time. Yes, undigested food does contain some water, but that's where we have our absorption, right? What else? What would be the primary component of our undigested food? Yes. But we don't call it that lah. You eat vegetable for what? What's the nutrient? Fiber. Fiber. Fiber is a very important component of our diet, but we don't get any nutrients or energy from it. What is it for? To help breathe means I want to have. Yes, to help prevent, right? To increase bowel movement, to aid with a smoother bowel movement. Okay, so we can talk about that later. Help with constipation. Okay. So in this topic, there's also a lot of emphasis on functions of the liver. Right? Okay, functions of the liver. Wait, have you all finished this topic in school already? No, so is it entirely new to you or halfway through? Somewhere doing already. Okay, so gone on to liver already? Yes, okay. Okay, so question. Where is your liver? Right side or left side?
Sorry, let me
Sorry, something left, is it? Liver is where? Liver is on your right. Liver is on your right. Okay, so if you can find your lowest rib, the rib cage there, the liver is halfway like a bit into the rib cage, but mostly outside of it. Okay, it is the largest internal organ. What is the largest organ? Very good, thank you. Okay. All right, so that's where the liver is, and it does an incredible job over there. Okay, so we can look at this. What are monosaccharides? The basic units of carbohydrates. So another thing about exam techniques is being able to understand the language of the question. When the question says, "Oh, monosaccharides are the basic unit of what molecule?" You need to be able to run through your mind. Oh, you're asking for carbohydrate, protein, lipid. I find the answer there. You cannot be what are monosaccharides the basic unit of sugar? You're thinking of the wrong things already. Right? Understand the language of the question and then scroll through for the correct group of answer. Amino acids, proteins and then the fats how. So this is where I want to bring to your attention this. What? The lymphatic system. Have you heard of the lymphatic system before? No, that one is still in the digestive system. The lymphatic system is an organ system by itself. And I'm going to tell you right now that this is actually a circulatory system. Okay, so since primary school, every time we say circulatory system, you think of blood, heart, and blood vessels. Okay, so this is actually in chapter 5, transport in humans. Heart, blood, and blood vessels. That one actually more specifically is referred to as the cardiovascular system. Sorry for next time, don't worry about that. Okay, so in your syllabus, have a cardiovascular system. So for now, the only thing you need to know about the lymphatic system is that it transports glycerol and fatty acids. Okay, glycerol and fatty acids. Okay, so in these 5 stages of nutrition in humans, the one that we focus a lot more on in upper sec here, the 2 A's, absorption and assimilation. Assimilation because we are looking at the role of the liver, absorption because we are looking at how the small intestine specifically is adapted for its function. What the small intestine does 2 very, very important things. Digestion and absorption. But digestion is just release the enzyme only. That's it. Correct now. So for absorption here, we are looking at and then
That's the force, and that's the noodles. That's that's cells. You know, like
yes, okay. So from to just look at this one, most important one. The large surface area to volume ratio in the small intestine is achieved by 3 levels of structures. But 3 things okay? 这很玩什么 largest to smallest.
force. Speedos. Wow, zero. What is that? Microvilli? Oh,
It's just microvilli.
oh, I thought it's that.
It's the microvilli on the epithelial cells. It's microvilli on the epithelial cells. I have so many more things I want to say, but don't have time. So let's continue. Okay. So bring your attention to this one again. What topic? What topic? Memorize topic number some more. What's the name of the topic? Movement of substances. Michelle, got learn not? Jody, other than diffusion and active transport, what is the third one, Jody? Osmosis describes the movement of water molecules. Thank you. Okay, so if you were to ask me the fundamental topics of bio MOS, movement of substances, is one of the most important ones. Because it will appear in many other topics. It will appear in digestive system, it will appear in transport in plants, it will appear in photosynthesis, it will appear in hormones, it will appear in no, don't have. Okay, but very important. Okay, because very simple topic, very simple topic. You will score answers for movement of substances as long as you remember definition. Your answer is just put a definition in, change the words here and there. Done. Okay, so go back and revise the definitions. All right. Okay, liver. What does bile do? Okay, wait, don't answer that. Does bile digest fat? Very important, huh? That's why we have the word AIDS over here. Gabriel, what word comes to mind then? Or rather, should come to mind then? Bile does what to fat? Gabriel? Emulsify. Thank you. Well done, Jaden. Jaden. Wait. We cannot just say emulsifies. What does it mean to emulsify? Break down fat into tiny. So how do we elevate our explanation from big to small? Say the from large fat droplets into smaller fat droplets. Again, flex your understanding there. So therefore, what we're looking for here, emulsification is where we have this is emulsified. What's the purpose of this? What do you say? Okay. Yeah. So the purpose of emulsification, the smaller something gets, the larger its surface area to volume ratio. We will no longer be using the primary school total exposed surface area, straight to surface area to volume ratio. Okay. The greater the surface area to volume ratio, the more effective the enzyme can function. What enzyme are we thinking of here? So what is this molecule? Fats, so what's the enzyme here to break them down? Lipase. Okay, lipase, So question. No bile means no fat digestion. True or false? False. Okay, then how? If no bile, what will happen to fat digestion? Slows down, a lower rate, takes more time. Very good. Okay, what happens to the person now? Hey, take note, if I cannot digest fat, I will not become fat. If I cannot digest, I can't absorb also. So what happens now? Tell me. I'm looking for an observation. What happens? Michelle, in a more eloquent way, please. Come, say. But in a more eloquent way, come, think of it. If I cannot digest the fats that I consume, what happens? Pass it out. This results in oily stools. Oh, oily feces. Oh no. So it's just that water slide out. Oh no. Oil slide out. A diarrhea is too much water. This is entirely different. It's oil. Well done. Okay, thank you for thinking about that. If you know, you know how this kind of thing. Okay, so these are things that we need to be able to expand upon as well. Right? We think about it, oh, if I cannot digest, then that's where we corrected Javen's thought there. Cannot digest, cannot absorb one. Right? So then where does it remain? Take the journey as the food particle, as I move through the alimentary canal. From not broken down, where will I end up? Alright, it's recommend the anus, so therefore all these tools there. Okay, wonderful, Michelle, thank you. Alright, so that's for bile. Store glycogen to regulate blood glucose because I don't want it to be too high. This very important point here, we cannot store except amino acids, so therefore we have to break it down to remove them. This was for Zheng's point there. The only alcohol that the body can metabolize and safely break down is ethanol. Okay, other alcohols, straight up poisoning of it. Okay? I will talk more about hormones next time once you have learned about more hormones there. Do you all know any hormones already right now? Insulin? What? Testosterone, what else? If you know insulin, you all should know the other one, what? Glucagon. Okay? Right, so throughout your syllabus, you learn a lot more as well. Okay, for now, must know insulin and glucagon. Okay, so I have prepared a couple of questions for us to work through, and I would like to just walk through with you together lah. Okay, so what I'm going to do right now is I'm going to present some guiding questions to some of you as well, so easier for us to understand questions. You will realize that how I like to do questions together is not just go straight to the answer, because that's my exam. We are not doing an exam right now. We are learning, we are revising, we are getting better. So I always like to expand on the questions. The first one is, I want to ask you, this molecule, what atom do I zoom into? Nitrogen. Because remember just now we started off today's lesson with atoms. And therefore, this belongs to a protein. So which of our four options is the protein? Okay, milk does contain protein, but in my option, milk.
Oh no, I saw it.
You are thinking of lactose is also known as a. Why you're thinking of that, right? It's because lactose is referred to as a. It's also called milk sugar. So you from lactose, you think milk, no milk or protein, there's no protein. Oh, it's the wrong chain maybe. Okay, right? No, this chain is factual, but not relevant to our question. Can? Okay? Yes, Marine, you're correct. Why? The answer amylase. Marine, share with us your explanation here. Why is amylase our answer? What is amylase? Can you tell me? Why? Jody, do you know why? How do you know amylase is an enzyme? Yes. And why do I want you to get to this point here? Is because if they give you an unknown enzyme, they never even tell you enzyme. They just say ACE. Wow, straight up, you will know it's an enzyme already. Can so learn that instead. Instead of memorizing your enzymes, remember how to name them. Then you can remember everything already. If you want to remember all your enzymes for all your 6 simple sugars, you learn 6 things. You must memorize 6 things. Why not just remember one thing? The front part, ASD. That's it. Okay? So ASD is enzyme, all enzymes are proteins, hence. Ta-da. Okay? Shanice, what is CHONS? CHONS refer to the 6 elements present in a protein. Oh, wow, that's very unique. Okay, Sam asks, what about cellulose? Because cellulose is found in plants, and plants absorb nitrogen from the soil. How do you think we can explain that?
So is it like a metal
Is what I shared with you just now. Cellulose found in plants. Plants absorb nitrogen. Not relevant.
Oh.
They are both facts. Yes, cellulose found in plants, factual. Plants absorb nitrogen, factual. Not relevant to our question. Not relevant to our question here. Can Sam? Okay, so what is the name of this molecule? Amylase is an enzyme. Enzymes are proteins. Proteins are made up of. Yes, amino acid. Amino acid. This is a basic unit. This is more amino acid. Again, let me elevate your learning here. This is the amino group. Anyone want to guess what is this part called? Yes. This one you learn in upper sec 4 camp. This is called a carboxylic acid. But for now, do we see why this basic unit is called an amino acid? Say no? Amino acid.
It's a second. What about amino?
The middle is always these two. This is the one that changes. Okay? Don't memorize this shape. Recognize. Like that. That's all. Amino and acid.
What's the section?
Section is this one. There are 20 different kinds because there are 20 different amino acids. Let's go. Food test. Let's go. Food test. Maybe wrong, eh? Can you share with us your answer for question 2, please? Sorry? A is for which test? Lipid is for which test? It's in the name here, the name of the test. Which one? Ethanol emulsion. Lipid, ethanol emulsion. White is present. Say? Yes, you are correct. For reducing sugar is which test? Absolutely. And yes, answer is A. Okay. Wonderful question here. The only thing that I want to help you with here, or that I need to help you with here, is to focus that the egg white suspension appears as a gray column. That's it.
So all the green parts.
Yes. The gray column represents egg white suspension. That's it. Oh wait, do you know what is found in egg white? Arisa, what do you think? Which biological molecule? Choose. Yeah, Zah.
What's the egg?
Please write down. This one needs to be common knowledge already. Okay, needs to be common knowledge already. Okay, egg white is about proteins. It's about proteins. So it's your body builder. Eggs. Protein. Build muscle. Actually, yolk is the unhealthier part of the entire egg. Yolk versus white. Yolk unhealthier.
So honorable man.
You know, don't go. Why? What do you mean?
But best, but I don't like the yolk.
Maybe she say best to taste leh. But it's only unhealthy if it's excessive. Like, you eat 1 or 2 times. Oh, okay, don't go and like one. Okay, so it's a protein. I tell you, protein. So if there's a protein, Jody, what is the name of an enzyme that digests? Jody, proteins. Thank you for making that correction there. It's not really a correction. I want to just share with you again. I always like to, again, remember just now I asked us about the amylase, the ASE, emphasize the ASE. So it's easier for your brain immediately to know there's an enzyme. If you protease someone, then your brain's like, what is this? Okay, so just go through a protease, lipase, amylase, sucrase. All this makes your brain immediately be able to draw the link. Okay, so you are already doing so much thinking. Help yourself do less. Okay, so Jody, again, you're not wrong to call it a protease. Okay, so for me, I just want to ensure that I can help you understand better. So when I say protease, you hear an ACE, the enzyme already. Okay. Hence, to answer this question, liver, mouth, pancreas, stomach, where got protease? Is stomach the only place for protease? But here no small intestine. How? Pancreas have or not? F. Okay. Liver don't have because factual not relevant.
Storage.
The liver does not release enzymes at all. Liver does not release enzymes at all. You think of that. Mouth there, why is mouth out? What's the name of the amylase? Okay, so for the amylase in your body, you need to specify you have 2 types. Why just name different only? No, it's not just name different. Salivary amylase. Okay, salivary so found in saliva. What's the other one called? Pancreatic amylase. Okay, form in the pancreas, but functionally, is there a difference between salivary and pancreatic amylase? They both break down starch, ah? Then how are they different? Yes, salivary amylase is the optimal pH of 7, neutral. Whereas pancreatic amylase, optimal pH of 8 or 9. So it's alkaline. So they're not interchangeable. So you need to specify. Okay. Wonderful voice. Thank you. So between the pancreas and the stomach, look at this. The only one that managed to break down the egg white was the one that was placed in the dilute alkaline. Alkaline is pancreas or stomach. Pancreas law.
Oh,
Pancreas, ah?
oh, because more greens. So
Broken down, ma. No need, not about blurry or not what. Half gray means that there on you. Okay, let's go. Experiment X was done at constant temperature of 37 for X. Y started at 37, then increased to 80. Can we immediately say the increase to 81 denature? Do we go straight to that? Why not? Just because I asked this way, right? Yeah. Okay, so here again, I want to impart a question analysis skill to all of you. When a question does not provide you an optimum temperature, and how? Because what? Let's say optimal 100, and one denature, right? All right, do you see that? So what happens when the question does not specify an optimal temperature for you? You take your body temperature. 45. Together, you know the hospital. Okay. For our body's enzymes, is ranging from 35 to 40. We find that range there. Okay. Okay. 40 to 45.
then I just
You know what? Hey, your body temperature is the same throughout,
start after this.
like 1 or 2 lah. 40, 45. Okay. So when because you don't need that specific number in your syllabus. So that's why we approximate it over here. We find 30 to 40, 35 to 40 is our optimum. Not inside yours. And textbook not ministry make mah. Who make one? Just the publisher, that's
Partial.
yeah lah. Okay, so what I want to say here is if the question doesn't specify any particular optimal temperature, go for the 37 range over there. Okay, so this means that the moment I increase above 37, so the denature, what happens to the graph? Now, when it denatures, it can no longer catalyze any reaction, right? So therefore, any products that have been produced already, should they decrease? Any products that I have produced already, what will happen to them? Stay constant. The product concentration should stay constant. So in experiment Y, there should not be any decreases. Product concentration goes down means less production. All I'm telling you is I cannot produce products anymore. So product concentration should not go down. So between B and C, what are we looking at? Who should have a higher? The difference is the endpoint. So how? Gabriel, do you think it's B?
Is it both referees?
No, only Y. Yeah. What do you think? B or C? Why is it B? So then so there will be less products produced at Y because the moment the enzymes denature, reaction cannot proceed anymore. So therefore, product concentration does not increase anymore.
So at 38 here.
It will slowly decrease. Remember the graph? Right? Okay, so it will not increase any further. That is one thing. Because it denatures, it will not increase any further. Can you understand this point? Michelle, are you sure? You always look at me like. Okay. We can also see that, what does this tell me? Whose rate of reaction was faster? Because the gradient's deeper. Why is faster? I thought denature. How to explain this? What about ultimate temperature?
More energy.
More energy. Remember, look at this. Gradually increase. I start at 37, I increase 38. Wow, more energy. Go 39, more energy. 40, most energy. 41, denature. Then we start to do this. Okay? Right? So there are two ways you can find B as our answer. Greater initial rate because higher temperature. Less products formed because denature. Awesome. Let's go. Okay, this one easy. Easy. What is it? Jody, help me with question 5, please. Sorry? Is it? Jody, why P? Thank you. Why were you confused with this one? What confused you, Jody? So read the question carefully. So like I said, answers are all in your head one. What I'm doing right now is to help you look through the correct category in all in your heads. Okay, wonderful. Careless, All right, question 6. I want you all to try and identify this one first. Based on the diagram, how do I know which biological molecule? Why?
So
So that's the same as memorizing amylase at that time. No, how to identify? What about it?
the arrangement.
Not arrangement. What about the shape?
Oh, the chain thing, is it?
What about it?
Don't know how to explain that one. It's like a lot of amino acids sheet together.
A lot of amino acids form what? So are you saying that this is a polypeptide then? So you're saying this is a fat molecule, is it? What do you say? Sorry, did I read you wrongly? What do you say? So polypeptide lah. Is it?
Because it's all the same thing.
Yes.
It's all close.
Yes. What's this?
Oh,
Polysaccharides are all made up of the same things. So in our question, the picture I give you, they are all made up of the same things. Whereas our proteins are made up of different amino acids. So in the diagram, they will give you like circle, triangle, square, like that, then like that on polypeptide because different amino acids. If they give you same shape, straight up, just go to carbohydrate. Okay? So therefore, has to be starch broken up by amylase. Please take note, this is a maltose molecule. Always remember, starch is digested by amylase into maltose. Please do not made the mistake of saying starch digested into glucose. So it is not a direct process, starch, maltose, glucose, amylase, and then maltase. Hey. What were the two words I presented to you earlier on about assimilation?
Success.
Use and store. So which of these is not assimilation in humans? Arisa, your answer is? Because? Yes. Question 7 is same as question 5. I changed the words only. Jody, the answer is C because? Answer is C because? Yes, Jody. I asked you because you were the one that explained to me question 5 just now. Yes. Yes, it's the same type of question. I changed the language only. Okay. Yes, look at that. Okay, so again, always learn the language of the question. Okay, language of the question very important, Okay, let's go. Now it's about enzymes. I love this question because you have to read all 4 statements. Let's go. Nadine, help us out, please. Question 8, you chose. Nadine chose D for Clarissa and Dorian. Michelle, do you agree? Huh? You agree? So tell me. Michelle, why is Alex incorrect? What's out? Wrong word. Not out, but above.
Wow.
Okay, so this first part. If I become cold, does the acting side change? No, it just becomes asleep. Thank you. So Alex wrong because of that. Jadine, Belinda is wrong because so not enough what? Not enough what? An inactive energy? Energy, very good. So Alexa's and Belinda's statements are both wrong because of the same explanation. So yes, Clarissa and Dorian are correct.
Just like average, like
Yes.
oh,
Awesome, let's go. What is this? This is a villus. Where can I find the microvilli? On the cell. Very good. So let me your eyes, please. Let me your eyes, please. So if I have to zoom in here, this thing these are the cells. The cell is not the microvilli. The cell has microvilli. Before we look at the question, can I have another question for you? What can I find here in the red region? What can I find here in the pink region? No?
Vacuum.
So vacuum. Where? Red or pink? Very good.
We need all this.
Hold on. What is in the pink then? What's the pink or the purple base? No, blood is the red already, ma. No. Oh, this place contains air. I want you to tell me what this place is.
Space.
Can't read this. Let me share with you this one word. Please make sure we learn how to use this. The word lumen refers to the space in a cylindrical object. So if I roll up my paper, I form a cylinder, right? The lumen is the space inside. The space inside. Your small intestine is essentially this. Correct? Right? So your food will flow through, get digested. It will have to be absorbed from the inside the lumen to the surrounding blood capillaries. Okay, so take note of that. So yes, we have no, because you use the words absorb from where into where. Okay. Can we identify P and Q first? Can we identify P and Q first? Arisa, help me identify one of them, please. No, I'll identify the thing first. P and Q, which one? Michelle, do you have another? Dial a friend. Arisa, dial a friend. Help ask for help, right? What is P? The answer is actually on the screen already. Be more specific.
Oh,
Chat, ma.
oh.
Right, P is the capillary, right? How do so dash network inside the villus. Okay? And therefore, Jaden, Q has to be the lacteal. The lacteal belongs to what system? The new one that I shared with you all today. The only time you will need to use it in your syllabus. Lymphatic system. Thank you. Okay? So, you only need to straight away remember this. And fatty acids and glycerols are going to be absorbed into the lacteals to be transported through the, you just said it, through the lymphatic system. Okay? Okay, so for the rest of the questions, right, we definitely don't have time to look at them, but right, don't back up first. Allow me to walk you through them a little bit here. Okay, like I said, I stress a lot on understanding the language of the question. So how can we look at this here? When we look at part 2, for example, describe how a name food test. Oh, immediately, what part of your brain should be looking for information for? It's going to be the for food test, straight up, right? Okay. I want to distinguish between the samples of impossible meat and beef. Are you going to compare one that has more and less or something? Or something that have, don't have? Definitely the have, don't have. You're going to go straight up to fiber or then how? Cannot test for fiber, right? Then how? How can we test for carbohydrates? Of the four, you can choose either Benefits or Benefits. Benedicts or iodine. Oh, carbohydrate what? Correct not? So your answer, your first line, you must take both have protein what?
Oh,
Okay? Okay? Alright, so that's it about the language there. Fiber. Say already. We talked about fiber just now, right? Okay, to help with bowel movement, not help with constipation. We help with bowel movement to reduce or prevent constipation. Okay? Okay? Okay, moving on. This is what I will refer to as a application question because look at the equation there. The only two words you recognize are oxygen and gas. Correct now.
oh.
The only two words you recognize are oxygen and gas. What? Then how? I lose already. Hey, no need. No. What did I say? Enzyme, right? Okay. So look at this. Yellowish tissue turns brown. This thing. Oh, yes, it is. But I don't need to know right now. The quinones are the ones that make it brown. Okay. I have made this question in a way to actually don't even need to use any of the words in the equation. It's just to put there for you to don't want to do really now. Okay, so right now you all have an advantage. The moment you all see these sort of things, right? Actually, the more weird stuff they put in the question, the easier to answer. Yeah, copy the weird stuff down lah. All you need to do is to read the question properly. Can be smart about answering your questions. If the question use this language, you use back for it lah. Confirm get a mark one. Okay, right? I want to bring your attention to lemon juice. Lemon juice. What was the lemon juice due to enzymes? Yes, straight up, you must say lemon juice contains acids or lemon juice is acidic. Okay. Then in the explanation, what word should we have in our heads? And hence, question says, could also by boiling. So what happened also? Again, this temperature and again, multinaturations. Right? We talked about appearances just now, right? In the food test page there. How would you put your answer here?
It goes from
Concentration. Appearance of apple flesh. What would the apple flesh look like after 20 minutes? Like your head. Not what? No. Okay, we cannot just say brown.
thing.
What's the word there? I highlighted for you. Yes, the remains and turns one. Thank you, Risa, for flipping through. The remains turns. Remember the food test? I highlight for you remains turns. So wait here, is it remain yellowish or turns brown?
But wait, what's
Remains yellowish.
Huh?
You all tell me acidic, you all tell me denature, then still can catalyze. Correct or not? Understand what I'm saying? so he's still confused. So this one acidic, this thing, denature already can This still happen?
That's oxidation.
Sit. Yes.
So oxidation.
Yeah. Yes, that's it. So the apple remains yellowish. Cannot. So what's the appearance now? What should we say? Remains yellowish. Thank you. Okay. Last but not least, this is just your small intestine one. So the reason I put this question over here is for you all to learn how to recognize photo micrograph as well, real life pictures instead of just my beautiful drawings. Okay. All right. Okay, so the answer keys are available online. Look at the Telegram chat there for the answer keys there. So that's all we have for today here. I hope that I have managed to help you pinpoint out how to answer and how to understand questions are. Like I said, the what is all in your heads already. Okay, so before we go, as with all my classes, right, I always like to invite you all to spend 1 or 2 minutes thinking through what we learned together in the 2 hours here. We're gonna share one point of reflection with everybody in class here. Okay, so as you're picking up, think about your reflection, but as somebody is reflecting, please respect them and listen to your reflection point to learn together. Whoever wants to go first, can go first. You don't have something? Not yet. Who want to go first, can go first. The sooner you go, the less likely your point gets stolen. Come, David. Anything that you want to reflect back upon in class here? Choose one concept, please. Cannot be vague one. Choose one thing. No, choose one. Yeah, choose one. Understanding the pH and temperature graph and how enzymes are affected. Thank you. Okay, you done? Chons, what does that tell me? Proteins, right? Proteins have chons. Okay, wonderful. Come, what about choice of vocabulary?
Using remains and terms.
Remains and terms, wonderful. Come, Jadine. What about the lymphatic system? Transports. Fats, very good. Arisa? Hydrogen and oxygen always in the ratio of 2:1 in what kind of a molecule? All carbohydrate. All carbohydrates, okay? All right, Marine? Sucrose is sweet girlfriend. Thank you for remembering that. Okay. Sun? What's the name of EA? Activation energy. Thank you, Gabriel. Assimilation is where the body uses or stores a certain nutrient. And Jody? What is ingestion? Enter and exit. Thank you. Michelle, have I asked you? Michelle? Must write the E and the A properly. Yes, very good. Thank you. Okay, so that's all the time we have for today. Thank you for the additional 5 minutes here. Just a reminder, the purpose we are doing this session at this timing here is so that if we have enough numbers, we can start a class at this specific timing here. So get in touch with the admin there. They'll get in touch with me, and then hopefully I can see more of your faces here and those online as well. And we can start a class learning together. It'll be much easier for us to start learning from Sec 3 rather than you scramble at Sec. Okay, I will have a longer timeline to help you build better learning habits, making sure that you understand everything more easily. Okay? Okay? Right, so think about it, check with your parents here, and I hope to see you more of your faces next time. Bye! Bye-bye.
Oh, sorry. Can I get one hot?
Hold on.